Well, good morning, everybody. Good afternoon, depending on where we're reaching you. My name is Shubhayan Sanatani, or Dr. Shu, as a lot of people seem to call me, and I am delighted to be the host of the Canadian SADS Foundation's webcast. Let's talk SADS live. And we are coming to you from Vancouver. And before we get going this morning, I wanted to introduce you to really one of the behind the scenes stars of our program, Evan Kruger, who quietly sits in the background and makes sure that I don't say anything rude, or he can bleep out things. And he mostly is providing wonderful technical support. Now, this is, of course, a part-time gig. He's in the process of graduating from the University of Toronto. So we're very grateful to Evan. I just wanted you all to put a face to the name, and he is a wonderful supporter to both the Canadian SADS Foundation and also in our wonderful relationship with the SADS US Foundation, who are, of course, close partners. Michael Ackerman started this concept about 120 webcasts ago, and we have collaborated with them in putting this together. So thanks, Evan, for making us all look good.
Great to be here. Great to help.
Trying.
Yeah. So we can just get your slides up here, and now I'll put myself down, and we'll get this started.
All right. Now I've lost my presenter view, but I will fix that momentarily. Am I sharing the right one? You never know. So just a reminder, this is where we come together to learn together collaboratively, and we can't necessarily dive into your particular medical care, but we love the engagement, and we've seen some great questions and comments in our chat, and so we're really here for our audience, patients, healthcare providers, and colleagues, those affected by SADS and those who just want to learn more about SADS. Now, I'm very excited to talk about this national auction. This is the first such event for the Canadian SADS Foundation, and the auction is not happening right now. The auction is happening in September 24th to August 1st. We're in the gathering stage. So if you have gifts that you think we would like to be to be promoting, are you an artist? Are you a photographer? Perhaps you're a travel agent. Perhaps you're connected to SADS because you have a friend or a coworker affected. So we are at the process now of really creating the inventory. And here's just some ideas. We have some of these items already. Maybe you want to donate a travel package. Maybe you want to just contribute a gift card, which is always welcome. So no amount is too small. And certainly check in at the SADS website, the Canadian SADS Foundation website. And this is going to be an important fundraiser. And why do we need funds to support the Canadian SADS Foundation? Well, good question. A lot of what we do is free, quotes free, lots of volunteer hours. Pam Husband, our executive director, has been donating freely of her time for a long time and works well beyond what she is compensated for. And that's a wonderful passion, and we want to expand our staff. We also want to work towards doing even more for our families. Maybe we can work towards a conference. We had a wonderful virtual conference, what feels like a long time ago, but I think it was just last weekend. And that's available still for viewing. There were about over 5 hours, and some of the talks were just outstanding, cutting edge talks by leading people in the field. There was a talk on pregnancy in inherited heart rhythm conditions, which is becoming more and more relevant now that some of our first patients diagnosed are reaching the age where they are considering families and having kids. So lots of content there from our virtual conference. Now, You'll remember that some of my Canadian fun, and to put a bit of a Canadian stamp on this, has been Canadian fun facts. And we've been quite medical and sciencey. We've had these very important Canadian contributors. Last month we had Banting and Best, which isn't really a heart story, but certainly diabetes affects the heart, and the invention of insulin in Toronto was a Nobel Prize worthy discovery. So today I went a little bit in a different direction, and we've got this very famous Canadian, and perhaps some of you recognize him already. Now, he is not a medical person, and does he really have a connection to SADS? Not particularly, except he did die suddenly at a young age, but under very different circumstances. So this is Tim Horton. Tim was not actually his first name.
I just need to interrupt quickly. I think
Yes.
we're going to present mode onto the thing because it's not on there, so they're not reflecting on the slides.
Okay, so should we swap presenter? Did that solve the problem?
Try to flip through the slides right now. Yeah, there we go. Yeah, you're good.
Okay.
Okay.
Do you have the present mode, or do
I
you
have it. I have it. This is good here. This is good.
All right.
Yeah.
All right. There you go. See, Evan just showing that he is keeping an eye on us. So this is Tim Horton, and died at the young age of 44, and a famous hockey player, won Stanley Cups, of course, and is now known because he started a chain of restaurants, very well known for donuts and coffee, and the famous Tibbetts, which occurred well after his starting the restaurant. And so it's a very successful franchise. So a Canadian fun fact about Tim Horton, who actually died in a car chase in his 40s. Now, on with the show. So I'm delighted to introduce today's guest. So Dr. Glen Tibbits is a long-term friend and mentor. He began his journey into the cardiac world quite a long time ago. When I started my first faculty position, and I had some research interests, people sent me across the street to the research building and said, "Why don't you talk to Dr. Tibbets?" And it's been a wonderful collaboration since that time. He began studying electrical and contractile properties of the heart during his MSc and his PhD, interestingly, at something called the Brain Research Institute. I don't know if that's because they mostly focused on brains, or you just had to have big brains to get in. Spent some time in Japan and still has connections and collaborators there, and then was eventually appointed to a position at UCLA and Seattle before moving back to his native Canada and being appointed at Simon Fraser University, which is where he and I began our relationship. So I'm delighted to introduce Glen Tibbits, and we've asked a special guest along that we're going to introduce shortly. So if we could bring Glen into the studio, that would be terrific.
Good morning.
Good morning, Dr. Sanatani.
Good morning. Good morning, Glen and Miriam. So I'll introduce Miriam. Miriam is a graduate student in Dr. Tibbets' lab, and so she has already completed an undergraduate degree and was so enthused about what she was finding. She wanted to learn more, and we're going to come back and talk a little bit about her master's, but I do want to take this moment to just give a real shout out. Part of her studies are supported by a family, and this is one of these heartwarming stories. The mother had a child who had long QT syndrome, and the child passed away a few years ago, and the mother felt that she really wanted to support long QT research, and so she reached out to Children's Hospital, even though her daughter had passed in her 40s, because that was where everything began for this family. So my thanks to the Harris family for supporting Miriam and the work that we do. So thanks for being in the SADS studio today, and I want to just, the title of our session today is about stem cells. And before we get going on to what can we learn from stem cells, I wonder if Glenn, you could just put out for us, what are we not talking about? I think all of us have seen, go to this lovely spa and get some stem cells injected into who knows what. Just differentiate what we're talking about and what might be for sale.
Can I show the slide that I think clearly shows what that is?
Definitely. So
Can you see that?
so we might not be in the presenter mode yet, but
we just need to be in the present mode, which will take over the screen, but we'll still see all of you.
Yeah. Okay. Thank you. So what we got into is quite different from what people are familiar with about stem cells being taken out of the body and maybe being injected into tissue damaged areas. And a lot of that stuff is very controversial. But what we use are called human-induced pluripotent stem cells, or IPSCs. And the way we start with this is we have patients, here's a family in SADS, and maybe the child has a genetic defect that might cause an arrhythmia. We can take a blood sample, and from the blood sample, this is where things change completely, is we're able to take the sample, grow the cells, and inject the cells with these magic factors called the Yamanaka factors. I'll explain why it's called that in a moment. And after about 4 or 5 weeks, these cells go way back to the future, and now they become stem cells from the patient, and they can become just about any cell we want. And of course, we're going to talk about heart cells, but our colleagues make pancreatic beta tissue, neurons, etc. And they're called Yamanaka factors because Shinya Yamanaka developed this technique about 15 years ago. But the amazing thing is, he got the Nobel Prize just 4 or 5 years after he developed this technique, and it's been completely transformative. So that's where we work, Shu, from the IPSC, or the human-induced pluripotent stem cells, as opposed to the stem cells that you were alluding to. Does that make sense?
Whoop, muted there. So yeah, very, very nice slide. We'll just leave it up for a moment. And important to point out that what we're able to do now is make these stem cells from a patient's own blood. When we were learning this stuff with Dr. Tibbetts, maybe 15 years ago or 12 years ago, we still needed tissue. So we used to take a little piece of, I have a little piece of my shoulder somewhere in a lab growing a new host for this show. And then of course, the cells then can be influenced to become the cells we need. And just to remind people, so a lot of the stuff that's in the lay literature and adverts and things about someone getting some stem cells just injected into their heart to heal the heart, that most of that stuff has not been very promising, because it's a somewhat simplistic view of how the body is going to accept foreign material. So anyhow, I've made the point, we're not going to be focusing on that today. So before we even go further, because I know we're going to get into some heavy science today, explain briefly, Miriam. So we've talked in this program, we've had a few sessions with some really excellent talks about genetics and variants, including variants that we don't know what they mean. So just tell us briefly, why do we need to work closely with scientists? Why can't doctors just take a genetic report and take it to the bank, so to speak? What is the value add of working closely with the functional assessment? I see Dr. Tibbetts is previewing your answer here with a very impressive slide. So Miriam, take it away.
Yes. So Dr. Sanatani, I'm sure you know our DNA is very different. 3.2 billion letters have to be arranged in a very specific sequence in order to make us us. And if you were to sit at a computer and you tried to type out 3.2 billion letters, you'd have to be typing at a speed of 60 words per minute, 8 hours a day for about 50 years. So that just kind of to put into perspective the complexity of what makes us a living organism. Now, of those 3.2 billion letters, you and I share 99.6% of them. They'll be identical. However, just that 0.4% difference between you and I is about 12 million letters. And within those differences is where we can identify variants that can cause genetic diseases and what we're going to be talking about today, arrhythmias. So oftentimes, and what you've experienced in clinic, is you may have a patient come to you after they've experienced some sort of event that could be syncope or cardiac arrest. And now what we want to understand from the basic science perspective is what in their DNA is causing that arrhythmia. And what may cause an arrhythmia in one individual because of some genetic variant may not be as pathogenic in another individual. And again, that goes back to that 0.4% difference between all of us. So what my research really relies heavily on is genome editing. Now, this is akin to a sci-fi novel, so to speak, because what we can do, what you see on the screen here, this big purple monster, kind of what it looks like, are really a pair of molecular DNA cutting scissors. So what we can do is use these scissors to go along those 3.2 billion letters and find where we have a variant that we are assuming is causing a inherited arrhythmia. And what we can attempt to do is either correct that variant and make it normal, so change that letter back into something that will no longer cause arrhythmia, or we can, when studying different diseases, we can create a variant and create a mutation to see what changes. So minute changes in DNA can have very large effects in the behavior of our cells. And that's really what genome editing and understanding the DNA of iPSCs allows us to do.
So lots of information covered there. I think I have 3.2 billion letters in my email inbox right now. But so you're talking, of course, about doing these things in iPSCs or in the induced stem cells. We're not doing this in people yet.
No, we are definitely not doing this in people yet. So iPSCs allow us a very personalized model. So just like Dr. Tibbetts mentioned, we could take a blood sample from you, Dr. Sanatani, and in our dish, we would have stem cells that are completely personal to you. They have your entire genetic background, and we are able to comb through that DNA and understand what makes you you. But no, we are not at the point of science yet where we can inject these big molecular scissors into a human or into any animal model yet. That is quite dangerous, and I believe there were some talks or conspiracies that people were or attempting to do this, and we're just not at that point in science yet.
I believe there have even been some people charged for doing experiments in that space. So, and of course, I'll just take this moment to remind our audience that not all of the conditions that we spend time in. So some of the conditions we understand the genetic mechanisms very well. Long QT syndrome is perhaps the best understood in that we have about an 80% positive genetic test rate when we have a typical case of long QT syndrome. So we can often find the genetic cause. Conditions like arrhythmogenic cardiomyopathy or even hypertrophic cardiomyopathy, the yield is lower. But again, it depends. I mean, if you have a very classic case of a condition, then we're more likely to find a variant that we then will know is the reason for the condition. Now, one of the reasons I lean on my basic science colleagues is that we often get variants of uncertain significance. And so I'm gonna ask our scientists if they could just show some of the type of work that you can do. So if I come to you, which I often do, and I say, I've gotten this result in the patient, and there's just not enough information. So take us through. I know you had a couple of slides. You had some heart cells, and you also had some electrical stuff. So let me turn it over to you to just show basically how cool the experiments are that you're doing and how much they are replicating the clinical conditions.
Great, thank you. This is work that was done about 8 years ago by Dr. Sanam, a PhD student in our lab, and a nurse took a blood sample from her, and then she used the Yamanaka factors to turn her blood into stem cells. And then after about 5-6 weeks, when they were stem cells, she converted her stem cells into heart cells. So what we have here is a Petri dish that's about 3.5 centimeters across. So there are hundreds of thousands, maybe millions of cells in here. After 10 days of trying to convert these into stem cells, the whole Petri dish started beating in just an incredible way. So that not only are there millions of cells, but they're talking to each other just like they would in an intact heart. So the electricity is going from cell to cell to cell, just as it does in the heart. But the electricity is something we measure. So here's the Petri dish, and we can see the electrical activity. And when we look at normal heart tissue, we can stimulate these just like a kid exercising from 55 beats per minute to 120 beats per minute. And you can see with healthy heart tissue, it's just beating normally. Of course, faster, but normally. But when Sanam introduced a variant that we weren't sure what it did, we found, the heart tissue was okay at low heart rates. But as soon as it went into moderate heart rates, like 75 beats per minute, we saw very serious arrhythmias. And then she found even slightly higher heart rates, kind of a disastrous consequence. So we didn't know about these cells, whether they were arrhythmogenic or not. So this technique gave us insight and perhaps how people got sudden cardiac arrest. Does that make sense?
Yeah, let's just pause on that graphic. And, it's a really important demonstration. In that top row, you can see what almost looks like an ECG. Some people have seen ECGs with the spike and the corresponding heartbeat. And then it's still electrical activity, but very different. So a kind of a somewhat normal and then a very abnormal, but early. And one can see how these, if you take this and you amplify it into a whole region of heart tissue, how exercise could lead to an arrhythmia, which is of course seen in so many of these conditions: hypertrophic cardiomyopathy, long QT, CPVT, etc. Nearly all the SADS conditions have some element of adrenaline driven arrhythmias. So going back, so I've come to the Tibbetts lab, and I say, it's important for me to figure this out. Now, I just want to digress for a moment. This type of work is very time consuming and very labor intensive. So I'm going to put a plug in here, because I hope there's a lot of members of parliament watching, but Miriam is a graduate student, and I'm pretty sure that the wage that she earns is probably well below the minimum wage, and they have not had a bump in what they get paid as graduate students for about 20 years. So I don't think there's too many sectors of the workforce where that's true. Now, I know everybody's a bit strapped for cash these days, but the grad students, the society has leaned on them heavily, especially the universities, without really compensating them. So if any of you have grad students in your lives, by all means, buy them a nice meal and write a letter to your MLA, because if Canada is going to be scientifically progressive, and that's extremely important for a country, we need our young, bright people supported. I think many of us, and I'm not going to get into the whole vaccine, anti-vaccine debate, that's messy stuff for some reason, but I think many of us were very grateful for the freedom that was quickly brought back in part by Canadian science. So anyways, that's my rant for this morning. It may not be my last rant. And I see Dr. Tibbetts has put up a tribute to 2 women in science. So I don't know if that's projecting right now or not, but I can see that. Maybe Glenn, are you sharing that? There we go.
Yeah, Miriam can talk about her heroes.
Yes, being a woman in science myself, and also we have to give tribute to these 2 amazing women, Dr. Emmanuelle Charpentier and Dr. Jennifer Doudna. They won the Nobel Prize in Chemistry in 2020, and it was the first time 2 women won this prestigious award. And it's nothing short of inspiring for their work in genome editing. So this inspiration to keep working, I'm sure these doctors, they began as research students, as graduate students, and to reach such a global stage is always on the back of my mind, and it's an inspiration to see them progress. But Dr. Sanatani, you are correct. Graduate students put in quite a number of hours laying this groundwork that can be translated into the clinical setting, and Canada does need to invest in this innovation, and I'm sure they understand the need to do so. And we just like to acknowledge, our genome editing heroes, so to speak, and to continue that science and what they've done so far.
Well, I know our colleagues at the SADS US Foundation have been very supportive. They've had an award. I had the privilege of adjudicating the award when I was the vice president of research for the Pediatric Electrophysiology Society. And what a great day it will be when the Canadian SADS Foundation has enough money that maybe they will offer a graduate scholarship or something like that. So anyways, that's what we're working for. We're working towards doing great things in science. Now, back to our patient. So we have a patient, you've demonstrated these abnormalities. Are you able to test different treatments in the Petri dish? Are you doing any of that kind of work? Because obviously we want better and more personal treatments.
Yes, definitely. Now, to speak to personal, that is exactly what iPSCs can be used for. So if I use the example of long QT syndrome, we can have acquired long QT syndrome. Now, this is drug-induced. Now, not so common because many drug manufacturers, there's much more strict testing to see whether a certain drug will cause long QT syndrome. But in order to test this, we use the IPSCs, we provide the drug, and after in our iPSC-derived heart cells, we can see whether this causes an arrhythmia. On the other hand, we also have inherited long QT syndrome. So this may be a child who presents in your clinic after they're playing tag at recess and suddenly they fainted, and now they're in a doctor's office and you're trying to figure out what happened. So the genetic components of long QT syndrome are many. There are 17 different subtypes. I'm focusing on long QT type 2. And now within that subtype, we have X number of mutations. So there can be hundreds of mutations that are different from individual to individual that will all cause long QT syndrome. Now, with those variations also come differences in how we can treat them. So one drug that may work for one individual in treating their long QT symptoms may not work for another. So again, using our iPSCs, we can reprogram a blood sample from a patient into stem cells, push them towards a heart cell lineage, and we can test different drugs. This is very personalized because now what we can see is how a drug is going to interact with that individual's unique genome. And we can see what drug is going to be best effective for that individual in treating their arrhythmia and going from that kind of messy signal that you see on the right of the slide back into a healthy left side signal.
Pretty amazing stuff. I think long QT type 2 is especially challenging because events occur both at rest and with activity. So it's one of the more common forms of long QT, certainly much more common than type 3. So 1 and 2 are the most common, but 1 is almost exclusively limited to exercise events. So very exciting to have these treatments. Now, I saw Dr. Dewar appear in our chat, and Dr. Dewar, of course, has a very interesting background as both a coroner in her past life, and then spent some time doing a PhD with you, and she was very much involved in some of the interesting work that we found in hypertrophic cardiomyopathy. So in the few minutes that we have, can you tell us a bit about some of your findings in hypertrophic cardiomyopathy? And we had Dr. Thomas Roston on last month, and he gave a wonderful clinical report on hypertrophic cardiomyopathy. I know that some of the findings that you've had in this space have been really eye-opening. So take us through a little bit of that, please.
Sure. So hypertrophic cardiomyopathy is relatively common, maybe 1 in 200, 1 in 500 cases, and it results in over contractility of the heart tissue. And that can cause the heart to grow bigger, hypertrophic, and become arrhythmic. And in terms of the known cause of sudden cardiac arrest in, say, elite athletes, and there's lots of unknown causes, but the hypertrophic cardiomyopathy variants can be a major risk factor. And those variants exist in these molecular motors that are shown here. And when we look at the actual molecular motors in the iPSCs, here they are right here. Each green line, between the green lines, is the molecular motor. And this is what Yasmin is doing in her PhD. And we can look at how they contract and see if they contract normally or if they're hyperactive, which is common to hypertrophic cardiomyopathy. This is a real problem for the patients when the heart is overly active, even under resting conditions. So one of the really cool things is that last year, in April, the FDA approved a drug which specifically inhibits the molecular motors so they're no longer hyperactive. It's called mavacamten, and is effective in treating these patients. It's not a cure, but it alleviates some of the symptoms, both arrhythmias and the growth of the heart, which is abnormal. So this is something we do a lot of, and we're trying to understand the molecular mechanisms by which this disease occurs and how we can prevent it from causing sudden cardiac arrest.
Fascinating stuff. And, the amount of time and work that goes into preparing even those findings is not to be understated. And we've talked a bit about mavacamten here also with Dr. Roston. Certainly a groundbreaking development in the treatment of hypertrophic cardiomyopathy, and approved in Canada a few months ago, I think, towards the end of last year. So now you're tempting us with one final teaser slide, I see, to have one more comment about some of the future. So tell us what you're showing us there.
Sure, this is really cool, and this is clearly part of the future of treating heart disease using 3D bioprinter. What's shown here is the 3D bioprinter in our lab, and we're able to put together different cells that make up the heart. So the cells that I've shown mostly today are the ones that are doing the contraction, but the heart is made up of many other kinds of cells that contribute to the heart function. And to create real heart tissue, you need to include those other cell types. And this field is just progressing at an unbelievable rate. One really cool group is in Pittsburgh, Adam Feinberg at Carnegie Mellon, and another group in Israel in which they are actually printing heart tissue with the valves, with the pulmonary veins and aorta, etc. And in the future, who knows, 20, 30 years from now, I think we'll be printing patients' hearts so that they don't need a donor, they don't need someone to die before they get a heart, and the heart is from their tissue, not foreign tissue, and they don't have to go on immunosuppression, which of course is problematic in itself, but then COVID comes along and they're immunosuppressed. So these are real issues. So in the future, we're hoping that we can print heart from the patient's cells, making stem cells, and replacing their defective heart with something like this. And this is so exciting. The field is expanding exponentially. But we still got a long ways to go.
All right. Well, I'm just noticing our time, and we'll just take the slides down for our last few moments here. I mean, thank you so much. You've shown us this. We could have called this session a show and tell of amazing science. It felt almost like science fiction. And I think it's important for our audience to understand a few things. First of all, that the conditions that we're talking about, when you get down to the DNA and the cell levels, they're actually quite a bit more complicated than just reading an ECG or prescribing a beta blocker. There's a lot of very complex physiology, even right down to the cells. So I myself in our program have a very important network of basic scientists, and most of the bigger programs that specialize in SADS have this. So certainly, at the Mayo Clinic and in Amsterdam and in Texas and all the ones, Vanderbilt, all of those programs have important partnerships, because we can't really we can't really advance the field without these important conversations and experiments. And we've seen a lot of this in the last year. We've seen more than in the preceding 5 or 10 years, I would say, with genetic therapies coming, new molecules, and certainly much more personalized assessment of variants. So I really want to thank Glen and Miriam for coming and teaching us a little bit about their world. And I saw one question in the chat from, will this work, talking again, I think about CRISPR-Cas9, will this work on any genetic variant? And I think it will. I see Miriam nodding her head. So lots to watch in this space. And I suspect, when we bring you back in 2 years to celebrate Miriam's graduation from her master's, we'll be much further along, and you'll have even more amazing things to show us. So I just really want to thank you both for coming and showing us just incredible right to the cell science. So thank you.
Thank you.
Thank you for having us, Dr. Sanatani.
It was great.
So, wow, it's I frequently feel like I feel like I need to go back to medical school to learn how medicine is being done, and the technology is moving so fast. And in part the computers, we're hearing a lot about artificial intelligence, and I think we're getting a bit freaked out, watching Mission Impossible movies, but the reality is the science is moving at a great speed. So we're just about out of time here. I do want to thank our partners at the SADS US Foundation and all the behind the scenes people, Alice, Lara, Aaron Waite, and Evan and Pam, who allow us to have these great guests educating us. And I'm looking forward to hearing a little bit from the audience, whether Tim Horton is a good Canadian trivia story, or if we want to go back to the medical nerd type things I've been showing. Just a reminder again about our auction. So our auction is very much in the gathering stage, and I think the more support we garner with your gifts and in your unique connections, the more exciting we're going to have that week in September, that September 24th week. So with that said, I'd like to thank you all for joining, and I look forward to next month. I think the theme next month is going to be Brugada syndrome, and we're very excited about a guest we're going to have who's done some really interesting science in Brugada syndrome, and looking forward to having you all back. So enjoy your summer, stay in the shade, and let's hope those fires get under control, which are burning in a lot of places. So thanks very much, and we'll see you next time.